Hello everyone, I'm Ho Jung-yu, a PhD student from University of Illinois at Urbana-Champaign. The topic that I will be presenting today is the effect of obstacle configuration on the transport of neutrally buoyant particles. Particle transport is a very broad topic which can be applied to numerous phenomena occurring in our surrounding nature. Among these, I would like to focus on microplastic pollution today as an application of particle transport, which is currently causing quite serious problems in aquatic systems worldwide. So as a start, what are microplastics? In definition, they are any kinds of plastic particles that have 0.1 to 5 millimeter diameter. They are exposed to nature from various sources, causing negative consequences such as entering food chain, carrying toxic chemicals, interfering water surface to block light and oxygen exchanges. Increased consumption of plastic and their slowly degrading feature are making problems caused by microplastics even worse. Thus, it became very important to study the transport of microplastics in aquatic systems and plan effective management strategies. Although majorities of current microplastic studies are focused on marine pollution, I want to point out that river is also a major source of microplastics, and if we successfully manage them early from upstream rivers, the damage caused by microplastics can be reduced. So in this study, we would focus on microplastic transport in freshwater condition, especially in unidirectional flow. So probably the major concern of microplastic pollution would be where are they? Would they be concentrated in certain locations? And yes, previous studies show that local hotspots of microplastics exist in nature, which are the areas requiring management plans, while these places can also be an effective target zone to capture microplastics. These references in the slide suggest that microplastics are frequently highly concentrated near structures inside the water, such as living organisms or artificial structures. So this study would also focus on hotspots of particles by installing artificial obstacles inside the water in laboratory scale. To wrap up, our research objective is to understand the transport and distribution of neutrally buoyant particles in unidirectional flow at different obstacle configuration based on surrounding flow structures. Specifically, we set 3 research questions. First, where would microplastics accumulate in unidirectional flow with different obstacle configuration? And in this presentation, I would only talk about the distance between 2 obstacles as a variable. Second, what is the most effective distance between 2 obstacles to capture particles in between? Third, are preferred locations of particles related to surrounding flow structures. So moving on, our study is designed for two-dimensional analysis of particle transport. All of our experiments were conducted at the Otto-Cavazzi flume, shown on this slide, which is designed for a small-scale unidirectional flow. So the straight section where we can see inside is the test section, and its length is 2 meters. Flow is driven by a disk located at the backside of the flume, which makes it possible to circulate identical particles inside the flume continuously. So we'll be analyzing both particle transport and flow structures, and to do that, hydrodynamics would be analyzed based on PIV, and particle transport would be analyzed based on PTV. So I'll briefly explain how PIV and PTV would be used in current research. So this is the actual recorded video, which shows the transport of particles, and to analyze their motion, this video should be saved as individual subsequent images. There are 2 steps in particle tracking velocimetry, and first step is particle detection. The center of particles are detected from each image based on pixel intensities, and these yellow dots are the locations of particle centers. Second step is particle matching, which is to match particle between 2 consecutive images. We see subtle movements between frame 1 and frame 2, and if we zoom in to this red rectangle, yellow particles are the ones detected at the first frame, which is called target particle. We pick one of them and locate it at the center of this interrogation window, which will be our new frame of analysis. Next, green particles are detected at the second frame, and those ones that are inside this interrogation window are candidate particles, and as the word indicates, these are the candidates that could be connected to target particle. So what to do now is to find out which one among candidate particles would be connected to this target particle. We do that by computing cross-correlation coefficients of image intensity matrices. So based on explained steps, we acquire full trajectories of particles that are detected from the video as this figure shows. Some are being captured in between the obstacles, while some just pass by above the obstacle. So next, now we want to analyze the flow structures based on particle image velocimetry. This video is what we would actually see in lab, laser shedding strong light on the targeted plane, and small PIV seeding particles moving inside the flume. Next, this video clip is what we would get from high-speed camera, which better shows flow structures. So again, we convert this video into consecutive images, and we use PIV lab included in MATLAB to get this result on the right. The algorithm of PIV is quite similar to PTV explained from previous slides, but the difference is that correlations are computed for interrogation window here, rather than individual particles to find out flow velocities. Now back to our experiment design, this slide shows you the experiment domain and our major variable. Actually, this research included other variables as well, but I'll only talk about the spacing between two obstacles in current presentation. So the distance would be varying from 1 to 10 L, where L is the length of the obstacle itself. And as a reference, the L is about 4 centimeter. And these obstacles at the bottom could be representing the submerged tree trunk or benthic organisms, and it can also be mimicking barriers and capturing devices installed at the riverbed in nature. The camera would be located at the side of the flume so that longitudinal and vertical movement of particles would be recorded, and light source would be located at the top to shed volume or plane of light to the flume depending on whether we are doing PIV or PTV. So this slide further explains the characteristics of particles applied in this research. We've chosen particle with 1 millimeter diameter, which is close to the median of microplastic particles. It has circular shape, which could represent bead or foam-shaped microplastics in nature. We're currently doing experiments with neutrally buoyant particles, which is in the range of common microplastics. So now we can finally talk about the results. Just by looking at this recorded video itself, we can observe that something interesting is happening in between these obstacles. Looking at this video on the left, you can see that particles are circulating within the spacing, which seems to follow the wake structures. At the right downstream of these two obstacles, particles are just residing there, having much less motion than other locations. Some particles are being captured for a long time, while some are escaping immediately. This video on the right shows that wake structures actually exist within the spacing as we have expected. So now our question are what would happen if distance between two obstacles is changing? And also how would we be able to quantify this phenomena? So we'll first start with PTV results. And this slide shows you particle trajectories during 1 minute overlapped on one image for each obstacle spacing case. As the distance increases, the shape of trajectories change, and when the spacing is larger than 4 times of length of the obstacle, circular shape starts to emerge. At closer obstacle distances, particles show random behaviors rather than having repeated trajectories. This could imply that there is a critical value of distance to generate strong flow structures and affect particle motion. When distance is further increased, new behavior is again observed where the circular motion is leaning toward the downstream obstacle And at the right behind of the first obstacle, random behavior of particle motion that was observed for short spacing is again appearing. So how would we be able to quantify the effect of obstacle spacing on the transport of particles? Here we suggest two possible indices, normalized capture ratio and retention time. As you can see from this left figure, we see that particles are being more captured in between obstacles as spacing increases until 5L and it starts to converge. Interestingly, this 5L is the point when particles start to circulate in between two obstacles from previous slide. So this figure on the right shows the retention time of particles. It was expected that particles would have longer retention time as spacing increases, since it would take longer time to move longer distances. It was true for particles that were passing by the obstacles above without being entrapped, here presented with this orange markers. And their linear trends is also very reasonable since the distance is linearly increasing. However, the result was quite different for entrapped particles, here represented with blue markers, and it had rather stepped function-like shape, retention time immediately decreasing right before the generation of circular motion of particles at 5L. So both trends of capture ratio and retention time imply that something is happening at spacing of 5L, and also spacing between obstacle itself is a significant factor to influence the transport of particles. So our next goal is to explain the behaviors that we have observed from particle trajectories, PTV, based on surrounding flow characteristics with PIV. I should first say that this is still ongoing research, and analysis of flow structures are not yet completed. Here I present only preliminary contours obtained via PIV analysis for specific spacing case 7L. Quivers drawn on the velocity contours and vorticity contour both show that flow structures are formed in between two obstacles, which has similar shape with particle trajectories observed from PTV analysis. Although this is not quantified with values, we still can expect that flow is affecting particle transport just by looking at these contours. Required future research would be to analyze how flow structures are varying for different spacing by quantification and how it is related to particle capture ratio and retention time. So this is the final page of my presentation. And as mentioned before, my research is still ongoing. So if you're interested in this topic and want to discuss anything and want to know further results, contact me anytime. I'll be happy to talk and discuss with you together. And thank you so much for listening. And thank you again.